Okay, in this video, we're going to learn how ERPs can be used to answer questions, in particular research questions. This presentation was made possible by materials from Pursue. You should check them out on pursueerp.com. So after watching this video, you should be able to explain why there's not a one-to-one correspondence between any particular ERP component and a neural source, explain the inverse problem, explain how to make cognitive associations with ERP components, be able to interpret what a difference in amplitude, latency, and scalp distribution mean, and explain the types of questions ERP studies can answer. So first, let's talk about making inferences about the neural source, right? We're just to refresh your memory, we're recording brain activity, and this brain activity comes from some neural source, right? A dipole, as we have discussed in previous. So in this example that you're seeing here, it's simple, one source. The reality is, if there was just one single source or dipole, then the time course of the neural source, the component, so we have on the left side, we have the time course of the source, hence the S1. And on the right side, we see the ERP, right? The observed component. So C1 here represents ERP component. So if there was one single neural source, the ERP would reflect that, right? That's what you would have. But the reality is, right, there is no one-to-one correspondence between ERP component and a single neural source because, right, the observed ERP waveforms are going to contain multiple sources of neural activity. And these multiple sources are going to overlap in time. Remember, they're going to capture all of the cognitive processing, right? So if we're talking about memory, it's going to capture perception. It's going to capture the memory part, but also sort of making a decision and making a response. So all of those cognitive processes are going to create neural generators and they're going to overlap in time. So it's not that simple, right? So there's some implication here, right? The timing and the amplitude of the ERPs are not necessarily going to match the underlying neural sources. So again, over here, let's suppose that we have different neural sources here, each indicated with a different color. And this is their time course that you can see those neural sources. But when you add them up, it gives three components, but the three components, the amplitude and the timing don't necessarily correspond to the neural sources, right? Similarly, the number of observed ERP components may not equal the number of neural sources, right? So it's not a one-to-one correspondence. Here's an example that sort of illustrates this possibility. So in the previous example, we saw that we had three sources right here on the left, and we ended up with three ERP components. But this doesn't happen all the time, right? Sometimes you could have more neural sources. Like in this case, we have four neural sources, and when they add together, we only have three ERP components. So that's possible too. You can't deduce the number of neural sources simply from the ERP components. Now, the other issue here is that changing in one underlying neural source may affect multiple ERP components. This is what we call sort of component overlap. So the implication here is that this can lead to interpretation problems, right? So let's consider this case here where you have three neural sources like we've been talking about, right? And we get our resulting ERP, and we just change one of the output of one of the neural sources. Well, notice what's going to happen is not only does the first component change, but also the second component changes as well, just from a change in one neural source. Here's another example. So let's change now, we're changing the second source, right? And you can see that it's going to change the second and the third components, right? So again, you have to be wary of component overlap. So changing just one could actually result in changes in sort of neighboring ERP components. Now we have what's called the inverse problem. So if we, like is indicated in the slide here, if we're given all of the pieces of the information, we can solve the problem. But the inverse problem is the reverse of this, right? There's an infinite number of solutions to come up with, in this case, the number 5. But the inverse problem exists because we don't know how many neural sources we have, their strength, their location, orientation, or their individual time courses, right? So it's very difficult to, if not impossible, to infer what the neural sources are just from scalp distributions. Now, despite this difficulty, people are attempting to solve this particular problem, and so there's some computational models, and generally what will happen is people will record high-density EEG from a lot of electrodes, like 128 or 256 electrodes, and then they try to do some mapping with complex models to arrive at a solution. And some other people are actually using functional MRI along with EEG to identify sources of brain activity. And this is a much better way to do this. But unless you're going to involve MRI, it's still somewhat problematic no matter how good your system is or how good your models are. A much better use of ERPs, in my opinion, is to use them to understand cognitive processes and the time course of those cognitive processes. So let's talk about that, how you make inferences about cognitive processes. This is really sort of the strength of this technique. So remember, we could collect behavioral measures, and these behavioral measures are useful, right? This is what the cognitive psychologist would do. For example, we could take response times, and this would tell us about the output of cognitive processing, how long it takes people to engage in a particular decision, for example, right? The beauty of the ERPs is that we can collect not only the behavioral data, but we can also get detailed moment-to-moment changes in brain activity over time. And one of the strengths of this is we could have the same behavioral outcome, like the same accuracy, the same response time, but have multiple ways in which the cognition unfolds to lead to that same outcome. The ERPs would tell us that because they would diverge. But still, this is tricky. How do we associate particular ERP components with some sort of cognitive processing? So again, like we saw with the neural sources, this is not easy, and there's not always a one-to-one correspondence between an ERP component and a single cognitive association. ERP components might be affected by multiple cognitive processes. So you might be studying one ERP component, and there might be multiple cognitive processes that it reflects. The number of cognitive processes might not observe the number of components, right? So you could have more cognitive processes and they don't necessarily yield the same number of components. And you could also have sort of the reverse as well. And a change in an early cognitive process might affect later ERP components, might spread or slow the stream of processing. So let's see a couple of more detailed examples of this. One example is the P3. So the P3 actually has different subcomponents to it that have been identified. So over here you see a trial sequence. S stands for standard, so it's a standard stimulus that's shown, and then all of a sudden there's a target where the person has to make a response. If we include some sort of distractor stimulus, one that's neither the standard nor the target, we'd see that we get the P3A, right? The P3A reflects sort of this novelty from this distractor stimulus. But when we shift from a standard to a target, we'd get the P3B, which is associated with different cognitive processes in and of itself. But they're both occurring right around 300 milliseconds, both have the same kind of amplitude, same kind of, they have a different scalp distribution. One is more anterior than the other. This is how they've sort of been dissociated, but otherwise they look very similar, and they are actually subcomponents of a similar ERP. So the cognitive associations also depend on the task and stimuli. This is something to keep in mind. So for example, you could be talking about N1. So we could have an N1 here and an N1 there, and you could be talking about a visual N1 or an auditory N1, right? And they're going to have different latencies, maybe some different amplitudes or different properties because the task and the stimuli are different. So you're going to have to be careful about defining ERPs, which is going to depend on the particular tasks and stimuli that you are using. So given all these constraints, is it even possible to interpret ERP data and make cognitive associations? It's tricky and difficult, and realistically, it's going to take place over multiple studies, not in any one given study. So let's talk about sort of an example to help illustrate this problem. Let's suppose that I give participants a task and say that adults took on average 600 milliseconds to complete this particular task. And then let's say I have another group of people complete the task and they complete it faster, 560 milliseconds. Without any other details, that difference is sort of meaningless. you would need to know what were the details, what was the task, what were the subjects, right? So what if I told you that on the left-hand side, this was older adults, on the right-hand side, this was younger adults, for example? That would tell you something about age-related changes in cognition. Or what if I told you the group on the left had a particular type of stimuli, and we changed the stimuli, made it a little bit easier to read so that that would speed up the processing, right? So the idea here is you got to go back to the experimental design to understand your data and understand your outcome, and the ERPs are no different in that respect. So we're going to look at how the physical characteristics of the ERP change along with our experimental conditions. And then we're going to make inferences based on both our existing knowledge, the experimental design and the conditions, what we've done, and correlating also any differences in ERPs to differences in behavior. And that is sort of the trick, is grounding it in behavior, which is not always possible depending on the design. And we're going to be practicing this when we read and discuss articles together as a lab. This is very tricky and it's difficult to do. So here are some common ERPs that you can see here, and you can find more descriptions of these online and in other research studies as well. Now, I do want to focus on ERP components that are most important to our lab. These are memory-related ones that we investigate. So one of the ERPs that we study is called the N400. Now, it gets its name because it peaks right around 400 milliseconds, and it's a negative ERP amplitude. Now, negative's plotted upward on this graph, so you have to wrap your brain around that. And the N400 is elicited a lot of times when you have semantically unrelated content. So, for example, you could present two related words like ring and emerald, and they would elicit the ERP that you see here in the solid line, right? If you then present two unrelated words like door and pinball, it's going to elicit the ERP that you see here in the dashed line. And you can see that there's a big negative peak, right? Remember, negative is up. Negative peak right around 400 milliseconds. Now, the difference wave shows you the difference between the unrelated and the related condition, and you end up with this dotted line here, and difference waves can be useful in showing different effects, like, for example, the N400 effect here. And if you look at the topographic map here, you can see that it has a pretty central distribution. Sometimes it's a parietal distribution, central. In this case, it's a central sort of right distribution for this particular effect. So that's one ERP that we look at. Another ERP that we look at is called the FN400, and it's similar to the N400, and I'll get to that in just a minute. But the F here stands for frontal, so it's a frontal negativity, right about 400 milliseconds. So typically we measure this 300 to 500 milliseconds is a typical interval that we use. And this appears in recognition memory studies because studied or familiar items elicit a more positive ERP amplitude than unstudied or unfamiliar items. So in other words, new or unfamiliar have a greater FN400 amplitude if you're talking about negativity. But typically, we plot ERPs positive upward, and we would say that studied items have a more positive FM400 amplitude than unstudied, or in this case, this would be missed items, right? So missed items are old items that are called new. And you can see in this topographic map that it has a mid-frontal topography that is characteristic of this ERP, and it's related to familiarity. It's a little more complicated than that, but there's another video that you hopefully have seen that talks a little bit about the ins and outs of the FN400 and its relationship to familiarity. The other ERP that we study is the LPC, or late posterior component. Sometimes people call it the parietal old new effect, and in this case, studied items are more positive than unstudied items. About 500 to 800 milliseconds is a typical window for that. And here you can see the topography is more left parietal, particularly for word stimuli, changes for picture stimuli. But this is a common one that we also look at depending on the research study. So some take-home messages. There are no one-to-one correspondence between the ERP component and the neural dipole. It's complicated. Similarly, cognitive associations or cognitive processes that are associated with ERP components, also similarly difficult. And you have to look at the total evidence and the experimental manipulations in order to make some conclusions there. So let's talk a little bit more in depth about interpreting differences in ERP components. So one way our ERPs can differ is in their amplitude, right? The height of the waveform. So like in this slide here, you can see sort of subtracting the two amplitudes. We saw that with the example with the N400 just a moment ago, looking at the amplitude difference, right? So what does it mean? An increase in amplitude typically indicates a greater neural response. We have to be careful though, because there's a lot of caveats to that. It could be good in the sense that it indicates more ability, more attention, but decreased amplitudes could also indicate greater efficiency. Also, you have to keep in mind that this depends, or the amplitude of the scalp waveform depends on the depth of the neural generator. So more positive doesn't necessarily mean more activity. It could actually mean the reverse, depending on the depth of the neural generator. So it gets quite complicated. The key here is comparing across the task conditions to interpret. That's what is most helpful. And also looking at maybe the scalp distributions can be helpful as well, which we'll talk about in just a second. Here's an example of interpreting ERP amplitude differences. So we could look at the N170, and the N170 happens in response to faces. And so the question is it just faces or any other visually similar items? So in this particular case, they presented faces and cars. And the result that you can see over on the right here is that the N170 amplitude was much greater, negative is plotted down here, for faces than for cars. So the interpretation here based on this evidence would be that it is more specific to faces, right? That it's related to processing those details. So interpreting latency differences, that's another one that can be used. We see that less used in memory research because it's slower occurring, more kind of tonic kinds of effects. You would see this more with perceptual kinds of research. But nevertheless, we'll talk about it here. So like in this example, you see two ERPs that differ, and they both have this early component here, but they might differ in time, where the latency is just sort of shifted in time a little bit by about 50 milliseconds. So what does it mean? It typically means a delayed or prolonged sort of processing. However, it could indicate that a new neural generator has come online. It might also indicate that an earlier process has sort of been delayed for some reason. So again, we have to be careful. Comes back to the interpretation based on the experimental design and other evidence, in particular other behavioral evidence. Now, one of the ways to really, and this is generally true for ERPs, is controlling for stimulus properties. This is particularly useful if you can keep the same stimuli, same people, but just change the experimental conditions. Then the ERPs can be mostly useful because you know it's not the stimulus or the people that are causing the differences. You can zero in on the fact that it's your experimental conditions which are causing the differences in the ERPs. So here's an example of a latency difference. So if we invert faces, it can affect the N170. So you can show upright and inverted faces. And we see that the N170 has a greater amplitude, but also a longer latency. See how it's shifted a little bit back in time. And so the interpretation here is that inversion would delay the perceptual processes for encoding faces. Another thing that we can do is we can interpret the scalp distribution. So here you have topographic maps, and you have young adults and older adults. And again, we have sort of our N400 kind of experiment. So you have positive minus neutral stimulus and negative minus a neutral stimulus. And you can see the scalp distributions differ for positive and negative information relative to for young adults, but maybe less so for older adults. So the scalp distribution change indicates some sort of change in the neural sources. So it could be, typically is a qualitative shift, like different neural generators of some sort, right? However, we can also look at the electrodes where the difference or the component is maximal. That can be helpful as well. And the other sort of caveat theory here is if you're analyzing these differences, you would pay attention to, some people would want you to range normalize your ERP amplitudes before comparing them to eliminate any main effects due to your task conditions. That's a little more of an advanced sort of thing, but it is something to be aware of if you're watching this video and planning to interpret scalp topographies. Here's another example from memory research where people have used differences in topographies to separate two ERP components and the cognitive processes that go along with those things. So the question is the FN400 different from the N400? They happen, and there had been a little bit of a debate that they were the same thing. Well, in this particular paper here by Bridger et al., they were able to sort of separate the two effects by separating the experimental tasks. So one task was a semantic priming task that typically elicits the N400, and you see that here in the posterior N400 topography, but then used episodic recognition, and that elicits a frontal FN400. Now these were the same people and same stimuli, so again, holding that constant and sort of separating the two tasks suggests that these are different ERP components and related to different cognitive processes. And we'll learn more about the theories behind that, and we're still testing that to sort of figure out exactly what cognitive processes map on to the FN400 and the N400 respectively. So let's talk about the purpose of ERP studies. We have some basic information that we could get, just identifying the component, right? Then maybe understanding what the component reflects in terms of its cognition, and then maybe even some cases apply it to sort of to better understand real world problems. So here's an example of just identifying a component. So Tim Kern in his 2000 paper presented evidence for the FN400. This is where it came from, this study. So people studied words, and at test, he presented some items that were studied, and it elicited both these studied items, elicited both the FN400, which you can see here, and also the LPC, which occurred a little bit later. But he also put on this test some words where the plurality changed. So like if you studied truck, it would be trucks. So this word wasn't actually studied, but it was similar. So those similar items elicited the FN400, but they didn't elicit the LPC. So this suggested that the FN400 existed, that it was separate from the LPC, and the interpretation here based on the study was that it was related to familiarity. So that was identified and examined in a host of studies, and eventually Voss, Paller, and colleagues across a number of studies pointed out that all of these studies that had looked at the FN400 had basically used words or pictures wherein familiarity co-varied with meaning. They all had meaning. And so they tested meaningless stimuli and found that the FN400 was eliminated. So on the basis of this, they argued that the FN400 didn't reflect familiarity at all, but it reflected instead conceptual fluency, or that is, meaning that could be conveyed by the stimuli that were being viewed. So we also reasoned that studying meaningless stimuli would change people's expectancies about what they would and wouldn't remember. And so what we did is we had people study meaningless abstract art. Now, under this idea, you shouldn't get the FN400 at all because if Voss and Paller colleagues are correct that the FN400 reflects conceptual fluency, that should be eliminated by the meaningless images. What we did, though, is we changed people's expectancies. We had a standard condition where we told people the same number of old and new items appear on the test. And then we had another condition, same people, same stimuli, these abstract arts, where they expected more old items on the test. And then we had a third condition where they expected more new items on the test. The reality was, on every one of these tests, the same number of old and new items appeared. Now, the key comparison here is we looked at, now these are just ERPs for old items, that is hits, and you can see that the FN400 changes with people's expectancies. And in particular, when you compare the old and the new, we can see that it has an FN400 or mid-frontal distribution that increases with a change in expectancies. So it may not be that the FN400 is eliminated when you have meaningless images. Instead, it may change people's expectancies about what kinds of experiences they're going to have on a test. So the story goes on. So the FN400 that we're talking about mark disease. And in this Guillaume 2012 paper, they found that the FN400 was present in healthy participants. So you can kind of see it here. You have old and different and new. They changed the background in their images, but the same person was presented with a different background. And you can see that the old and different are more positive than new in this particular graph of control subjects. But for subjects that had schizophrenia, it wasn't present for any of the conditions at all. It was eliminated with schizophrenia, and it suggests that the disease has some deficits in recognition memory. So this is a way in which the ERPs can be applied to real-world kinds of issues, in this case, patients with disease. So some take-home summary: differences in amplitude, latency, and scalp distribution reveal different cognitive processes. Research can clarify the components themselves, help understand cognitive processes, and maybe even be applied to broader questions.